So inflammasome is composed of three molecules. One is NLRP3, not like receptor protein. Another is caspase-1, and the other is AAC, that is adapter protein. This NLRP is actually sensor, and this caspase-1 is actually in inactive formation. After getting some different signals from pathogen, like it can be from bacteria, like pore-forming toxins from S. aureus, Clostridia, flagellin protein, peptidoglycan fragment, RNA or DNA from bacteria, and virus derived RNA or influenza M2 protein, or fungus derived different sugars or hyphae, and protozoa derived hemojoin can induce this inflammasome to form. So these molecules actually form a supramolecular complex. And also you can see sterile activators like self-derived molecules and environmental derived molecules like skin irritants, UV radiation, alloy particles, silica, asbestos, alum, and cell-derived, in this case ATP, cholesterol crystal, any crystal like urate crystal can develop this inflammasome. In this slide, I will explain the function of inflammasome. What happens? Pathogenic bacteria or extracellular any ATP that induce NLRP3 called sensor, and AAC, adapter protein, and caspase-1 in inactive enzyme form this NLRP3 inflammasome. There are different type of inflammasome, but this NLRP3 is very distinguished. What is the actual main function of this NLRP3 protein? It actually produces from caspase-1 inactive enzyme to caspase-1 active. So the main function of this inflammasome to produce active caspase-1 protein. This caspase-1 play an important role. Innate signals from toll-like receptors or other PAMP activate pro-inflammatory cytokine like pro-IL-1 beta or IL-18. This cytokine, when produced, are in inactive in form like insulin and glucagon. This type of protein actually called zymogen, because when they are produced, they are in inactive form, but after enzymatic cleavage, it becomes active. So caspase-1 at first was inactive in condition, but by forming NLRP3 inflammasome, it becomes active, and that caspase-1, active caspase-1, cleave the pro-IL-1 beta, that is inactive in form or non-functional, become functional, and this interleukin can induce acute inflammation. The inflammasome, the activation of NLRP3 inflammasome, which processes pro-IL-1 beta to active IL-1, is shown. Inflammasome with other NLRP proteins function in a similar way. Various PAMPs or DAMs induce pro-IL-1 beta. So the ultimate result is induction of inflammation. This inflammation is actually sign of 4 situations called redness, swelling, warmth, and pain. So where there is inflammation, this phenotype will happen. One is redness, swelling, warmth, and pain.